How do we reconstruct phylogenetic trees from data? Well, if we take the historical overview again, one way of thinking about evolutionary histories is to simply dig into the ground, find fossils, and then from the progression of fossils through the different geological strata, start thinking about the evolutionary changes that may have occurred over time. And one famous example is that of the evolution of the horse, illustrated here by fossilized hoofs collected in different geological strata, where we have the gradual evolution from a typical five-toed animal up through different forms to the version of the horse we have now, that's essentially just walking on a single digit. By looking at fossils like this and directly seeing changes, it's possible to start forming ideas about the evolutionary histories that have occurred through time. However, as I mentioned in the start of the course, fossilization is a highly improbable process. There are not that many fossils that we could form the full history of all animals on the planet. Therefore, it is fortunate that it's also possible to build evolutionary trees using only present-day data. The main idea in all of these methods, and this is actually true of any method we will look at in the course, distance-based, parsimony, maximum likelihood, and Bayesian methods, what's in common to all of these methods is that they form the phylogenetic tree by trying to group the organisms that share the most traits. I've tried to illustrate that on this slide. So looking at this slide, if we start in the upper left corner with the fish, the fish is different from the rest of the organisms on this slide in that it has no lungs. The frog, the lizard, the cat, and the chimp all have lungs. The fish does not. Now, the simplest explanation for this state of affairs is that we know, first of all, that if we go far enough back in time, then all of these organisms will have had a common ancestor. We know these are all derived from a common ancestor. So the simplest explanation must be that this original ancestor, this original population, split into two. One branch eventually led to the fish, the other branch led to the other animals, and at some point this other branch, lungs, evolved. After lungs evolved in this ancestor, every single descendant of that ancestor now also have lungs. This is a monophyletic group, this is a clade. They share this trait, that they've inherited from their ancestor. Okay, if we now look at the rest of the animals on this slide, then the frog, unlike the rest of them, have no claws. The fish, of course, also has no claws. Again, the simplest explanation is that this original ancestor split into two populations. One of them eventually led to the frog. The other eventually went up here, evolved claws, and every single descendant after that ancestor now also have claws. So that's why they share that feature also. Looking at the rest of the animals, the lizard is different from the cat and the chimp in that the cat and the chimp have fur and mammary glands. They are mammals, as we say. Again, the simplest explanation is a split, one branch leading to the lizard, the other branch leading to the present-day mammals. So you can see this illustrates, in fact, the idea in all of the methods we will use, even though we will use different statistical or philosophical ways of extracting or of using that information. But really, the idea is always the same. We try to group animals that share the largest number of these so-called derived traits. This is the idea. This is how you can use present-day data to reconstruct a phylogenetic tree. When we compare animals like this, I said all of these animals have lungs, all of these animals have nails. It's important that we only use features that are what we call homologous. Homologous meaning they have actually derived from a common ancestor. It's the same sort of thing that's derived from a common ancestor. This slide showing the bone structure in various limbs of human, dog, bird, and whale illustrates that nicely. You can see quite clearly, I think, that they all have the same bone structure, a single upper arm bone, two lower arm bones, and then some bones in the wrist and some other bones at the digits there. And illustrated in color, you can see the corresponding parts. So homology is important for comparing animals. You need to compare the same thing in the different animals, or it doesn't make sense. This is sometimes problematic when you look at morphological data. For instance, this example At the left, I have an illustration of a so-called red-headed vulture. That's a vulture living in the Old World, Africa and the Indies and Europe. On the right, we have a turkey vulture. It's a so-called New World vulture. Like the condors, it's living in the Americas. Superficially, these animals seem quite similar. They both eat dead animals for food. They are bald. They have these crooked beaks, and they have large wings, and they look alike in many different morphological aspects. And therefore, it was originally believed that they were probably closely related and that they formed a single group. However, when we started getting molecular data, it very rapidly became obvious that they were not each other's closest relatives. Instead, the Old World vultures, like this one, were more closely related to birds of prey, such as hawks and falcons and eagles, while the New World vultures were more closely related to storks. The reason they look so similar is that they have the same lifestyle, they are under the same selective pressure, and therefore they end up finding, in essence, the same solutions to the problem that they're trying to solve. In this case, one important problem is if you eat a dead animal that's infected with bacteria and rotting away, then apparently it seems to be an advantage not to have feathers on your head where all of this rot and maggots and what have you can stick. And you also need a crooked beak in order to be able to rip up the gut, etc., etc. So the reason they look alike in this case is that they are living under the same circumstances. It's the same phenomenon if you compare a shark and a dolphin. They look very similar. The shark has a vertical tail, the dolphin has a horizontal tail, but their body shapes are quite similar. But again, that's not because they're each other's closest relatives. So convergent evolution can sometimes fool you if you look at morphology, but not so much if you look at molecular data, mostly because you have so much of it. It's therefore important that when we compare things, we want to look for homology. Homology meaning similar traits that have been inherited from a common ancestor. We do not want to look for homoplasy, homoplasy being similar traits that have evolved independently, maybe by convergent evolution, that are not caused by these animals having common ancestry. An example of homoplasy, the wings in these animals, the pterosaur, the bat and the bird, they all have wings. As it turns out, these wing structures have evolved independently. The bird uses one set of digits to spread out its wings, the bat uses another set of digits and incidentally supports the wing using some of these digits. The pterosaur used still a third set of digits. And the wings were made from quite different materials also. So these three wings, though they look similar, they have evolved separately. They are homoplasies. When we talk about molecular phylogeny, homology, homologous characters, mean columns in alignment. If you have an alignment of DNA sequences like you have here, 4 species, A, B, C, and D, then you start out by aligning the sequences to find the similar regions, and this is how you figure out which nucleotides are homologous. So for instance, you can easily see that there are conserved positions here. Then in the column labeled 1, we have GG, TT. This means that at some point in time, the ancestor of all of these sequence or all of these animals had some nucleotide at this position. Maybe it was a G, maybe it was a T, and then at some point a mutation occurred, leading some of them to differ from the rest. The same with the others. So in molecular phylogeny, when we talk about a homologous character, we talk about a single column in an alignment.